Hello everyone. In this video, we are going to investigate the effect of light on gas exchange in an aquatic plant. Okay. Now, in previous video, we have discussed actually about the effect of light on gas exchange in normal plants. Okay. We have seen that during daytime, whenever light is present, plants, they perform photosynthesis as well as respiration. Okay. So during daytime, plants, because of the presence of light, they photosynthesize at faster rate compared to the process of respiration. And hence there is net input of carbon dioxide and net output of oxygen gas. But during night time, because of the unavailability of light, plants, they only perform respiration. And hence during night time, there is net intake of oxygen gas and there is net output of carbon dioxide gas. So in this video, we are going to investigate the effect of light on gas exchange in an aquatic plant. And in order to perform this experiment, you need an indicator which is called hydrogen carbonate indicator. And we are using this indicator because this indicator is highly sensitive to pH of the solution. Okay, hence this is also called pH indicator. Okay, so hydrogen carbonate indicator is used because it is pH sensitive. It is also known as pH indicator. Okay. Now, the purpose of using this hydrogen carbonate indicator is, I've already told you, this is pH indicator, and hydrogen carbonate indicator actually shows the concentration of carbon dioxide gas in water. Okay. So what happens is, carbon dioxide gas, whenever it is produced, okay, whenever carbon dioxide gas is produced in the process of respiration or any carbon dioxide gas which is present in the air, so this carbon dioxide gas dissolves in water. Okay, and after dissolving it in water, carbon dioxide gas forms a very mild acid which is known as carbonic acid. Okay, so carbon dioxide gas dissolves in water and forms a very mild acid which is called carbonic acid. Okay, and this carbonic acid makes the water slightly acidic. So this change in the concentration of carbon dioxide gas in the surrounding water of aquatic plant can be detected by hydrogen carbonate indicator. Okay, hydrogen carbonate indicator is pH indicator. When carbon dioxide gas dissolves in water, it forms a mild acid which is called carbonic acid. So if more carbon dioxide gas will dissolve in water, the water will become more acidic. Okay, because more acid will be formed. And if less carbon dioxide gas dissolves in water, the water becomes alkaline, or it's still acidic, but it's lesser acidic than the previous one. Okay, so in next slide I'll just show you the color of hydrogen carbonate indicator at different concentrations of carbon dioxide gas. Okay, so remember, the original color of the hydrogen carbonate indicator is orangish red. Okay, so if you look at hydrogen carbonate indicator, you can make out that it's orangish red in color. Okay, or it is reddish orange in color. Okay, now whenever hydrogen carbonate indicator shows reddish orange color, red or reddish orange color, that means the concentration of carbon dioxide gas in that particular water or that particular solution is equal to the concentration of carbon dioxide gas in atmosphere. Okay, in atmospheric air. So in atmosphere, the carbon dioxide concentration is 0.04%. Okay, so whenever the concentration of carbon dioxide in any water or solution will be 0.04%, the color of hydrogen carbonate indicator would be red. Okay, red or orange, or sometimes it is reddish orange. Okay, if the concentration of carbon dioxide gas in water or in any solution goes more than atmospheric level. Okay, remember here I'm telling you about more than atmospheric level. The level of carbon dioxide concentration in atmosphere is 0.04%. Okay, so if carbon dioxide concentration in any solution goes above atmospheric level, so the color of hydrogen carbonate indicator will change to yellow. Okay, so remember this, if carbon dioxide concentration goes above atmospheric level in any solution, so the color of hydrogen carbonate indicator turns yellow. Okay, and when it will happen? It will happen in case of plants only if the rate of respiration is higher than the rate of photosynthesis. Okay, because when the concentration of carbon dioxide will increase, when the rate of carbon respiration is higher, because in the process of respiration oxygen gas is consumed and carbon dioxide gas is released outside. But in the process of photosynthesis, carbon dioxide gas is consumed and oxygen gas is released outside. So as the rate of respiration is higher than the rate of photosynthesis in a particular plant at a particular moment, so more amount of carbon dioxide gas will be liberated outside the plant or released outside the plant. So as the carbon dioxide concentration is increasing in the surrounding water of the plant, what will happen? The indicator's color will change to yellow as it's shown here in the table. Okay, if the carbon dioxide concentration in the water around aquatic plant goes below atmospheric level, okay, so what will happen? The color of the hydrogen carbonate indicator will change to magenta. First it will change to magenta and then it will change to purple finally. Okay, so remember, this is how the hydrogen carbonate indicator helps us in identifying or helps us in understanding the concentration of carbon dioxide gas in the in any aquatic solution. When carbon dioxide concentration is equivalent to the concentration of carbon dioxide in atmosphere, then the color of hydrogen carbonate indicator would be red or orange. Okay, if carbon dioxide concentration increases above atmospheric level, so the color of indicator would be yellow, and if the carbon dioxide concentration decreases below atmospheric level, so the color would first change to magenta from red and then finally it will change to purple from red. Okay, and when the color of hydrogen carbonate indicator is red, that means when the carbon dioxide concentration is equivalent to the carbon dioxide concentration in atmosphere. Okay, that means at that time the rate of photosynthesis is equal to the rate of respiration. And if the color of hydrogen carbonate indicator changes to magenta and then finally to purple, that means the rate of photosynthesis is higher than the rate of respiration. As the rate of photosynthesis is higher, so more and more amount of carbon dioxide is being used up in the process of photosynthesis. Hence, carbon dioxide concentration decreases and the color changes to magenta or purple. Okay. In order to perform this experiment in laboratory, you need 4 test tubes or 4 boiling tubes, tube A, B, C and D. Okay, and in all these 4 test tubes, you actually have to add 20 ml of hydrogen carbonate indicator. Okay, it's always written as centimeter cube because centimeter cube is actually a unit of measurement. Cube refers here volume. So 20 ml of hydrogen carbonate indicator you have to add in all of these 4 test tubes. Okay, now in test tube A you have not taken any leaf because this is your control tube. So we will just compare the color of hydrogen carbonate indicator in rest 3 test tubes with the color of hydrogen carbonate indicator in test tube A. Okay, so no leaf has been added in this test tube because it is a control tube. Then in test tube B you have to place a leaf in the tube. Okay, so in test tube B we have taken a freshly plucked leaf from a plant. Okay, in test tube C you have to place a leaf in tube and wrap this entire test tube in aluminum foil to block any light which is falling on the plant. Then in test tube D you have to place a leaf in a tube and wrap in a gauze to wire gauze. Okay, wire gauze to allow partial light. Okay. And finally, you have to just close the mouth of all these 4 test tubes with the split cork. Okay, and leave all these 4 test tubes in light for 30 minutes to 1 or 1.5 hours. So here is a picture which represents the experiment to be done by us. Okay, you have to take 4 test tubes A, B, C and D. In all these 4 test tubes, you have to add 20 ml of hydrogen carbonate indicator. And you can check out that the color of the hydrogen carbonate indicator is orange. It can be red also. Okay, so red or orange, or it can be reddish orange. In test tube A, no leaf has been placed because this test tube acts as a control test tube. In test tube B, C and D, 1 leaf has been added in each test tube. Okay, and then the mouth of the test tube is closed with wire gauze. Test tube C, before placing it in bright sunlight, we have actually covered this entire test tube with aluminum foil so that no light has been provided to this leaf. And in case of test tube D, the entire test tube is covered with a wire gauge. Okay, so this wire gauge actually provides partial light to this test tube. So after placing all these 4 test tubes in light for 30 minutes to 1 hour, what happens is you will get the following result. So in test tube A, the color of the hydrogen carbonate indicator will remain same, because inside this test tube A there was no leaf. So even when the light was falling in this test tube, no photosynthesis could take place. Okay, so no photosynthesis, no respiration, hence the concentration of carbon dioxide was equal to the atmospheric level. Okay, so the color of hydrogen carbonate indicator remained same. In test tube B, it was placed in light. Okay, fresh leaf was present, so this leaf has photosynthesized in the presence of bright sunlight. Leaf has performed photosynthesis. Okay, now what happens is, as this leaf has performed photosynthesis, so the carbon dioxide gas which is present in the air in this test tube has used up by the leaf for the process of photosynthesis. So the concentration of carbon dioxide gas has been decreased, and as the carbon dioxide concentration is decreased, so the color of this test tube is changed to purple. Okay, so purple color indicates that leaf has performed photosynthesis, and because of photosynthesis, the concentration of carbon dioxide in this test tube is decreased. Test tube C shows that leaf is present. Okay, this test tube was kept in bright sunlight also, but what happened is this test tube was covered with aluminium foil. Okay, so this aluminium foil has blocked any light which was falling on the test tube. So no light has reached leaf because the entire light was blocked by this aluminium foil. So as no photosynthesis was performed by this leaf, but respiration was going on. Okay, so here the rate of respiration was very high than the rate of photosynthesis. Okay, there was no photosynthesis at all because of the absence of the light. So as the rate of respiration was very high, so more and more carbon dioxide gas was released because of the respiration in the leaf cells, and hence the color of hydrogen carbonate indicator in tube C is changed to yellow. So yellow color indicates the carbon dioxide concentration is higher than the atmospheric level. Okay, purple color indicates the color of carbon dioxide concentration is lower than the atmospheric level. Okay, and this orange or red color indicates that the concentration of carbon dioxide in this solution is equivalent to the concentration of carbon dioxide in atmosphere. In tube D, this leaf has performed photosynthesis because of the partial light which was entering inside the test tube and falling on the leaf, and leaf has also performed respiration. Okay, so in test tube D, the rate of photosynthesis was equivalent to the rate of respiration. Okay, so as the rate of photosynthesis was equal to the rate of respiration, so the concentration of carbon dioxide gas was maintained in this test tube D. Hence, there was no change in the color of hydrogen carbonate indicator. So remember, in test tube A, there was no live specimen. So this test tube A was acting as a control. Okay, there was no photosynthesis, no respiration in test tube A. In test tube B, rate of photosynthesis was higher than rate of respiration. Okay, so the color has been changed to purple because as the rate of photosynthesis was higher, so more amount of carbon dioxide gas was used up or consumed from the air present inside the test tube. Hence the color of hydrogen carbonate indicator has changed to purple. In test tube C, the rate of respiration is higher than the rate of photosynthesis. No photosynthesis was taken place because of the aluminium foil which was blocking any sunlight falling on the leaf. Then in test tube D, the rate of photosynthesis is equal to the rate of respiration. You can just go through this table. In test tube A, no leaf was present. It was acting as a control. Initial color of indicator was orange and final color was also orange. That means no respiration or photosynthesis taken place. So there was no net movement of carbon dioxide gas. Okay. In test tube B, leaf was shown to the In test tube B, leaf was shown to the bright sunlight. Okay, so the initial color of hydrogen carbonate indicator was orange and the final color is purple. Okay, this is because due to the presence of bright sunlight, rate of photosynthesis was higher than the rate of respiration. Hence, there is more intake of carbon dioxide gas. So the level of carbon dioxide was decreased inside the air. Okay, hence color has changed to purple. In test tube C, leaf was wrapped in aluminium foil. Actually, the test tube was completely wrapped in aluminium foil. So no light was given to this test tube or this leaf. Okay, so initial color was orange and final color was yellow. That's because the carbon dioxide concentration in the air of test tube C. This is because the carbon dioxide concentration in the air which was trapped inside test tube C has increased because here the rate of respiration was very high. No photosynthesis was taken place. Okay, so there was only the net release of carbon dioxide gas. And in test tube D, this entire test tube was wrapped in a gauze. So gauze has provided partial light to the leaf. So rate of photosynthesis was equal to the rate of respiration. Because in all three cases, respiration is going on. Okay, but no photosynthesis could take place in test tube C, and in test tube D, photosynthesis has taken place, but at normal rate. Okay, at a rate which is equivalent to the rate of respiration in that particular leaf. So there's no net movement of carbon dioxide gas, and hence the color of hydrogen carbonate indicator remained same. Okay. Now I'll just show you a small video on this experiment. This experiment is a little bit different than the experiment which we have studied in the PowerPoint presentation, but you will be able to understand the concept clearly. Okay, and you have to perform this experiment by taking 4 test tubes, and these test tubes are labeled as A, B, C, D, and in all these 4 test tubes you have to add 5 or 10 ml of hydrogen carbonate indicator. Okay, and you can make out here in the video the color of hydrogen carbonate indicator is reddish orange. And in test tube A you have placed a fresh leaf. In test tube B you have just placed this small wire gauze, and you have added carefully wood lice or small insects. These animals are added because it will perform respiration. So you'll be able to understand the color of indicator. In test tube C, you have taken animal plus leaf, both. And in test tube D, you have not added anything. So test tube D here is acting as a control. Okay. So remember, I hope you have understood. In test tube A, only a fresh leaf has been added. Okay. Only a fresh leaf has been placed. In test tube B, there's no leaf, only 2 wood lice. That means only the animal is placed here. In test tube C, animal is placed as well as leaf has been placed. Okay, and in test tube D nothing has been placed, so this test tube D is acting as a control. Now you have to just place all these 4 test tubes for 30 minutes to 1 or 2 hours in bright sunlight. Now you can make out, after keeping the test tubes in front of bright light for few hours, the test tube A, okay, this is your test tube A, the color has been changed to purple. Test tube B, the color has remained same, that is reddish orange. In test tube C, the color has been changed to yellow. Okay, now what happened is, in test tube A the color is purple because in test tube A fresh leaf was placed. Okay, so this leaf, when it is placed in front of bright light, obviously this leaf will perform photosynthesis. So in the process of photosynthesis, entire amount of carbon dioxide gas which was present in the air, which is trapped in this test tube, was consumed in the process of photosynthesis. So leaf has consumed entire carbon dioxide gas which was present in the air here in this test tube in the process of photosynthesis. So as the concentration of carbon dioxide was decreased because of the process of photosynthesis, because it was consumed by the leaf, so the color of this hydrogen carbonate indicator was changed to purple from this reddish orange. Okay, in test tube B, actually what happened is, Now you can make out here, this test tube D is control. Okay, this test tube D is control. So the color of hydrogen carbonate indicator here is same like the original color. In test tube A, because this leaf was placed, so leaf has performed photosynthesis and hence carbon dioxide concentration is decreased inside this test tube. Okay, so the color of hydrogen carbonate indicator has been changed to purple. In test tube B, this wood lice was present. Okay, so this wood lice has actually consumed this wood lice. In test tube B, wood lice is present. Okay, so this wood lice has actually performed respiration and carbon dioxide gas was released in the air. So as the concentration of CO2 was increased in the air, so the color of hydrogen carbonate indicator has been changed to yellow. In test tube C, if you remember, we have actually placed a leaf as well as this wood lice. Okay, so this leaf has actually performed photosynthesis and wood lice has performed respiration. So the rate of photosynthesis and respiration was equal in test tube C. Okay, hence the color of indicator would be orange red. So I hope this experiment is clear to you guys. In next video, we will discuss about mineral requirements in plants. Take care till then. Bye-bye.